Good morning, viewer. Being the chair, here's a breaking news. Yeah, Sinai health researchers have discovered that a critical transition in early human development is regulated not by our own genes but by DNA elements called transposons that can move around the genome. The remarkable discovery challenges our previous understanding of these elusive DNA segments, illuminating their impact on human development and diseases. People tend to think of transposons as akin to viruses where they hijack our cells for the sole purpose of propagating themselves, says studies senior co-author Dr. Miguel Ramalho Santos. Yeah, senior investigator at the Luenfield Tenbaum Research Institute, that's called LTO, part of Sinai Health, and professor at the Department of Molecular Genetics at the University of Toronto. But here we have discovered that these elements are not more genomic parasites but are essential for early development, said Dr. Ramalho Santos, who holds the Canada 150 Research Chair in Developmental Epigenetics. Their study, published October 15 in the journal Developmental Cell, indicates that transposable elements are critical to ensure that human embryonic cells progress normally through early development rather than going back in time. The researchers focused on the transposable elements known as line 1 for long interspliced nuclear element 1, unlike our own genes, which compose less than 2% of our genome. The line 1 elements comprise a staggering 20% of the genetic material in our cells. Some of line 1 elements can amplify and move around the genome, inserting themselves in new locations because they spread of their own accord in a way that can disrupt normal gene functions. This has earned them the moniker of selfish DNA. For years, scientists believed these elements were mostly harmful, occasionally wreaking havoc in the genome and contributing to a variety of diseases, from hemophilia to neurological disorders and cancer. Dr. Juan Zhang, a senior co-author and postdoctoral fellow who spearheaded the research, initially found it intriguing that line 1 RNA messages are abandoned in early embryo, and RNA message molecules are transcribed from parts of the genome that are active, indicating that line 1 elements are switched on this critical early stage. If transpositions are bad and dangerous, why do we see them active in the early embryo? This is an embryo that's just beginning its formation. Any dangerous insertion into the genome at the point is going to be propagated throughout the rest of the development of the individual, said Dr. Chang. Being this exclusive report.